Hello students, today we can see the topic lipids. Lipids are also one of the biomolecules which is present inside the cell. Lipids are organic compounds which are formed of carbon, hydrogen and oxygen. Lipids are water insoluble, that is they do not dissolve in water. When we put a drop of oil or any lipid in water, you can see it remains as a separate layer without mixing with water, isn't it? So lipids are water insoluble. The lipids can be simple fatty acids or glycerol. A simple fatty acid consists of a carboxyl group attached to the R group. R group, or we can say it as the long hydrocarbon chain. Okay, a simple fatty acid consists of carboxyl group attached to the R group or a long hydrocarbon chain. The R group or the long hydrocarbon chain may be methyl, ethyl or higher number of CH2 groups. Okay, the R group can be methyl, ethyl or higher number of many number of CH2 groups. And the number of carbon atoms will be 1 to 19. So the number of carbon atom present in the simple fatty acid could be 1 to 19. For example, palmitic acid is having 16 carbon including the carboxyl carbon, and arachidonic acid is having 20 carbons including the carboxyl carbon. This is palmitic acid, it is having 16 carbon atoms. This is arachidonic acid with 20 carbon atoms. The physical nature of fat is determined by the chain length of fatty acids and whether they are saturated or unsaturated. So the fatty acids could be saturated fatty acid or unsaturated fatty acid. So what is saturated fatty acids? Fatty acids which do not have double bond between the carbon atoms are called saturated fatty acids. So they are having only single bond between the carbon atoms. Okay, so what is saturated fatty acids? The fatty acids which do not have double bond between the carbon atoms are called saturated fatty acid. Example, palmitic acid, stearic acid. Palmitic acid is having 16 carbon and stearic acid is having 18 carbon. Okay, now what is unsaturated fatty acid? Unsaturated fatty acids are having one or more double bonds between the carbon atoms. Okay, unsaturated fatty acids are having one or more double bonds between the carbon atoms. For example, oleic acid, linoleic acid and linolenic acid. Oleic acid is having one double bond, linoleic acid is having two double bond and linolenic acid is having three double bond between the carbon atoms. Okay, this is the structure of oleic acid. This is linoleic acid. This is linolenic acid. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids are both glycerol and fatty acids. Here the fatty acids are found esterified with glycerol. That is, the chemical bond formed between glycerol and fatty acid is called ester bond. That's why we say that the fatty acids are found esterified with the glycerol. So here three hydroxyl groups of glycerol molecule unite with the carboxyl group of 3 fatty acid to form fat or lipid. Okay, 3 hydroxyl group of glycerol molecule unite with the 3 carboxyl group of 3 fatty acid to form fat or the lipid molecule. And most of the lipids are esters of glycerol. That is why these are called as glycerides. Okay, and these glycerides can be monoglycerides, diglycerides or triglycerides. What is monoglyceride? Mono means single. So here one fatty acid will be esterified with the glycerol. That is called the monoglyceride. And what is diglyceride? Di is 2. So 2 fatty acid will be joining with the glycerol and it forms diglyceride. And triglyceride means 3 fatty acids will be esterified with the glycerol. Okay. This is the structure of monoglyceride. Here you can see one fatty acid is esterified with the one glycerol molecule. This is diglyceride. Here, two fatty acids are found esterified with one glycerol molecule. And this is triglyceride. Here, three fatty acids are found esterified with one glycerol molecule. Okay. These are also called as oils or fats based on their melting point. Oils are having low melting point and they remain as liquid even during winter. Fats are having high melting point and they remain as solids. Example for oil is gingerly oil and example for pure fat is tripalmitin and tristearin. Some of the lipids are phosphorus or phosphorylated organic compound attached to it. So these are called as phospholipids. So what are phospholipids? Lipids which are having phosphorus or a phosphorylated organic compound in them are called phospholipids. And these phospholipids are commonly found in the cell membrane, and an example for phospholipid is lecithin. So lecithin can be found in the cell membrane. Then in the neural tissues means the nerve tissues, it have lipids which are having more complex structures. Okay, so one of the lipid with a more complex structure is cholesterol, which is found in the neural tissues. Okay, so this is about lipids. This is the structure of cholesterol. So that's all about lipids. Next we can see about nitrogen bases. In the living organisms, there are a number of carbon compounds which are having heterocyclic ring structure. So this nitrogen bases are carbon compounds with a heterocyclic ring structure. And the nitrogen bases are adenine, guanine, cytosine, uracil and thymine. So these are all having the heterocyclic ring structure. In the previous classes we have seen about amino acid and lipids. So they are generally having linear structure, but this nitrogen bases are having heterocyclic ring structure. Now these nitrogen bases are of two types, that is purines and pyrimidines. What are purines? Adenine and guanine are said to be purines, and cytosine, uracil and thymine are called as pyrimidines. This is based on their structure. Okay. Purines are adenine and guanine. Pyrimidines are cytosine, uracil and thymine. Here you can see the structure of adenine and guanine. These are purines. They are having double ring structure. Now these are the pyrimidines. Cytosine, thymine and uracil are the pyrimidines. And here you can see they are having a single ring structure. Okay. Where the nitrogen bases are present, they are present in the nucleic acids, DNA and RNA. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. Now the nitrogen bases present in DNA are adenine, guanine, cytosine and thymine. And the nitrogen bases present in RNA are adenine, guanine, cytosine and uracil. So instead of thymine, uracil will be there in RNA. Okay. When this nitrogen base combines with the sugar, it forms the nucleoside. Nitrogen base, it can be any one of this, adenine, guanine or cytosine, thymine, any one nitrogen base. When it combines with the sugar, it forms the nucleoside. The sugar which is present in DNA is deoxyribose sugar, and the sugar which is present in RNA is ribose sugar. This is the structure of ribose sugar which is present in RNA. It is a pentose sugar, that means it is having 5 carbon, and in the second position you can see OH group is there. That's why it is a ribose sugar, and in DNA it is named as deoxyribose sugar because in the second position one oxygen is not there. That's why it is named as deoxyribose sugar. Okay. So when a nitrogen base combines with the sugar, what is formed? Nucleoside is formed. Example of nucleoside is adenosine, guanosine, thymidine, uridine, cytidine. Okay. So adenosine, if nitrogen base is adenine plus sugar forms adenosine. Guanine plus sugar is guanosine. Thymine plus sugar is thymidine. Uracil plus sugar is uridine. Cytosine plus sugar is cytidine. So these are the examples of nucleosides. This is the structure of nucleosides. Here you can see adenosine, adenine plus sugar, cytidine, cytosine plus sugar, guanosine, guanine plus sugar, thymidine, thymine plus sugar, and uridine, uracil plus sugar. Okay. Now when the nucleoside combines with the phosphate, it forms the nucleotide. Otherwise we can say when the nitrogen base combines with the sugar and the phosphate, then it forms the nucleotide. Okay, nitrogen base plus sugar plus phosphate forms the nucleotide. So here the phosphate will be seen esterified with the sugar, that is between sugar and phosphate ester bond will be formed. Okay, thus a nucleotide will be formed. And example for nucleotide is adenylic acid, guanylic acid, thymidyllic acid, uridylic acid or cytidyllic acid. So these are the examples of nucleotides. Okay, if adenine is the nitrogen base, then it will be adenylic acid. If guanine is the nitrogen base with the sugar and phosphate forms the guanylic acid. Thymine with the sugar and phosphate forms thymidyllic acid. Uracil with the sugar and phosphate forms uridylic acid, and cytosine with the sugar and phosphate forms cytidyllic acid. So these are the examples of nucleotides. This is the structure of nucleotide present in RNA. Here you can see a nitrogen base is there, sugar is there, and the phosphate is there. These three join together to form the nucleotide. Okay, then same way here you can see the nucleotide of a DNA, again on nitrogen base combines with the sugar and a phosphate. So here the sugar is deoxyribose sugar. Okay. These nucleotides are the building blocks of nucleic acids, DNA and RNA. That means DNA and RNA are formed of nucleotides. Okay. So many nucleotides will join together to form the DNA or RNA. So we can say this DNA or RNA as polynucleotide strand. And this DNA and RNA act as genetic material. They function as genetic material, that is they can transmit the genetic information from one generation to the other. Okay.